So, once again we are here with a series of molecular biology lectures. We had learnt about how nucleic acids are formed, and then we had seen how the DNA is organized, that is how it is wound over histone octamer and how it is finally compressed into chromatins, two sister chromatids and in chromosome. So, it is almost 10,000 compression, but actually book to book it varies, and in some books again it is being mentioned now, and of course things do change with time, because this molecular biology is something that is growing ever since it has been taken up. So, DNA is compressed 8,000 times. If we go for the packing ratio, how it is zipped, this very folder can take you that how the fold is, the DNA is being zipped into the chromosome and the chromosome being kept in the nucleus. So, the naked double helical DNA, if we consider it as one unit, then 10 nanometer fibril of nucleosomes. After this DNA, naked DNA is wound over the histone octamer. What is that known as? Beads on string formation. Then 30 nanometer chromatin fiber of superhelical nucleosomes, that will be in the ratio of 40:60, and finally when we come to the condensed metaphase chromosome loops, it will be 8,000 times packed. Fine. So, this is how it is compressed so much that such a big DNA is kept or accommodated in a very submicroscopical organelle, that is the nucleus. Now, this who helps the histones to come forward and form this beads on string formation or whatever. See, if you see at nature, nothing is absolutely useless or worthless, nothing is unsystematic, nothing is disorganized. For everything there is a fixed rule and regulation. That is why when we are ourselves disciplined, things run very fast, you are a more successful person. Likewise, if we come to this, you will see that histone on their own do not form dimers, then tetramers and octamers, and like that the DNA will not come and wrap around it. Rather, some proteins are involved in this. So, they are the histone chaperones. So, if you remember from your protein classes, chaperones means the old ladies, they were accompanying young, in old British era, they used to accompany young ladies to meeting someone or the so called dating sessions. So, they will sit aside having a watch over the couple. So, that was the real purpose of chaperones. Likewise, here also there are certain proteins like histone chaperones, they help in winding of this DNA over the histone octamer. So, they will guide the octamers, histone octamers to the site of DNA, and once they get attached to the histone octamer, the histone chaperone proteins, they will gradually separate out. Now, there is a term known as phasing. What is this phasing? This phasing is, these nucleosomes have affinity for specific sites of DNA, but why it is so, the basis is not known. That means some task for you people to do in the future. Now, what is the significance of this phasing? The phasing is likely related to both to the relative flexibility of a particular nucleotide sequences to accommodate to the regions of kinking within the supercoil, as well as the presence of other DNA bound factors. That means this is a platform where the different processes of DNA binding proteins, they will come and bind, or they are the kinking, they are the regions, the weak regions where the kinking of the supercoil will start. So, this phasing is also clinically significant for us. Now, this transcriptionally active or potentially active regions have little different nucleosomal structure. As we have seen, the nucleosomes, the free DNA takes 1.67, in Harper it is written 1.75 helical turns, then a 30 base pair of free DNA, that is the linker DNA, and another histone octamer. So, it will be like going clockwise, then in the next histomer it will be anti-clockwise, again clockwise and anti-clockwise, like that the histones are wrapped around by DNA. Now, these areas of chromosomes, if you see the these chromatid bars, so there will be certain areas which are transcriptionally active. That means these are the regions which can get converted, or they will produce the desirable mRNA which will produce the desirable protein. Suppose there is a call from the periphery of our body that we need more of globin for hemoglobin synthesis. So, the globin gene will be activated at the DNA level. So, DNA level the gene is activated and it will give rise to its specific mRNA, from which mRNA the protein globin will be synthesized. So, this transcription does not occur in the entire DNA, it will take place sparsely at certain places, and how much is the gene content actually, which are the expressed regions, that will come see in the subsequent slides. So, these transcriptionally active parts, regions, they have some altered nucleosome structure. So, which are very susceptible to the DNA's action. So, here the DNAs, they are very active and they can make single strand cuts. Then another term comes hypersensitive sites. Now, they are the interspersed regions between active chromatin. They are even more susceptible to nucleus action, giving access to the DNAs probably. So, these are all newer terms that you will be coming across, and usually these are present just upstream of the genes that are being expressed. So, these are the hypersensitivity sites. They are even they are present apart from this active chromatin and little upstream, so that from there the transcription actually starts. Okay. When we will go to transcription, it will be more easy to understand. Then we had discussed the other day about euchromatin and heterochromatin. So, U means true, where the different processes will take place, and heterochromatin usually the condensed areas, which will be found usually in the centromere or in the telomere region. So, these are the denser areas where it is little sparse, it is little loosely present, then it is the these are the dense areas are the heterochromatins, and the sparse areas are the euchromatins, where different processes will take place. Now, this there are some inactive regions which are high in methylated deoxycytidine DNA content. Okay. That is important from research point of view, if you say so, and they have lower levels of activating covalent modifications and high levels of repressing histone PTMs, that is post translational modification. So, the histone formation at these regions are little slow. So, what happens? the euchromatin is replicated faster than this heterochromatin. Usually what idea we have, once the DNA starts replicating, means it starts from one end and it gets covered for the entire DNA at one go. No, it is not for the eukaryotes, not for the higher organisms. It is true for the prokaryotes. It begins from one side and it ends at the other end. But for higher organisms like us, our DNA replication will begin at different places. So, what we have seen is euchromatin is replicated little faster than the heterochromatin. Now, there are other two terms, constitutive and facultative genes. Now, constitutive genes, they are always inactive, and constitutive areas in fact, they are always inactive and condensed and found in centromere and telomere, which are usually remaining as it is. What function they have, particularly since they are silent, they form the structural formation, and facultative, at the time of need they will transcribe, that is they will synthesize or they will give rise to the mRNA and the subsequent proteins, and they are seen at the regions of euchromatin. So, euchromatin and facultative segments are not same, but they have a little similarity, you can say. And if you go for the sex chromosomes, that is XX, so one X permanently remains inactive till gametogenesis, and it becomes transcriptionally active during early embryogenesis. So, it is basically this one X which is remaining inactive, that is the constitutive one, it is behaving constitutive, and at the time of need it becomes facultative as well. So, these are the terms that need to be understood. Then another term is polythene chromosomes. When multiple cycles of DNA replication takes place without separation of daughter strands, usually there are multiple copies of DNA joined together, and there is a huge large chromosome that is seen, known as polythene chromosome. This phenomena is seen in case of Chironomus and Drosophila. Fine. Another term you might have come across very frequently in your +2, now these things have come up. So, telomere, it is involved both in aging as well as cancer. So, telo, as the name suggests, it is end. So, at the end of the chromatid bars we have these telomere regions, and these are Tg rich regions. So, this is the characteristic feature of telomeres, and usually there are 5' TTA GGG 3' repeats. I had told you that 5' to 3' polarity always will be maintained whenever we read the sequence, it is from 5' to 3' or 5' to 3'. So, these sequences, the repeats in variable numbers, they will be present in the end, that is the telomere. So, they can extend up to several kilobases. Now, this telomeres, when the DNA is completing one cycle of replication, it loses the end part of the DNA, not the entire DNA will be completely synthesized or doubled. So, the end part is due to this telomerase, one enzyme is there that will synthesize this telomere part and maintains the length of the DNA. So, this telomerase, what it is? It resembles somewhat to a viral reverse transcriptase enzyme. Reverse transcriptase means RNA to DNA. So, this converts the RNA to DNA and synthesizes the DNA part, and it maintains the telomere length. So, just you can imagine if telomere or the entire DNA is intact cycle after cycle. So, probably it is maintaining our youthfulness. So, if that telomere shortening once starts, that is telomerase is deficient, it is not able to synthesize this telomere, then we will land in aging. Yes, that is me. So, I may look like this, and telomere lengthening leads to cancer. So, if there is unchecked activity of this telomerase, then there may be cancer. Now, what is the implication of this? Many drugs or therapies can be found out which will target this telomerase enzyme. So, it can work as a very good anti-cancer therapy. So, this aging etc., aging and cancer both are very important to the entire world. No one wants to become old. Now, you cannot understand because you are in your prime youth, you do not have that feeling how the body aches or how the knee aches after walking for a few kilometers. Now, you are at the prime youth having utmost vitality. So, when once you cross 40 or 45, you feel the pain or weakness, then you start hankering for how to prevent aging. And what makes your skin very supple? You have read it in the carbohydrate chemistry, and that is the basis of the entire cosmetic industry also nowadays. That is the GAGs, the glycosaminoglycans. They are trying very desperately to maintain youth, but youth can be maintained by modifying your lifestyle, like I had told you earlier. Now, you will think I can booze, I can smoke, I can drug. What will happen? Nothing will happen. Over years, in another 10 years, you will see the effects. These are all slow changes, and how these things happen, that we will see under DNA replication and repair. Okay. Cancer is also just an altered form of that. So, how you can prevent this? By having a disciplined life, waking up early, doing little meditation. Now, this meditation has no link with religion. People usually merge religion and lifestyles very foolishly. Actually, meditation is neither the sole property of Hinduism or anything. Yoga is not property of Hinduism, it is a way of life, and you must acquire these exercises and this meditation, that is concentration. So, maybe someday we will have a session on this also. Now, coming back to our DNA, the molecular level things, what is happening? Only 1% of DNA codes for about 1 lakh proteins that we require for our body, be it structural or any kind of function that we have, it is all proteins and proteins. So, we have such a huge DNA, yet only 1% of DNA codes for 1 lakh proteins. And it is seen that only 25,000 protein coding genes are present. If you see the entire length, it is the genes, the chromosome is 1.5 into 10 to the power 8 base pairs. Now, this small chunk we have enlarged to see the gene cluster. Suppose 20 genes are there, it is just 1.5 into 10 to the power 6 base pairs. Then one gene if we are taking, this is the sequence, and from this gene when we get the mRNA primary transcript, it becomes from 2 to the power 2 into 10 to the power 4 base pairs to 8 into 10 cube, that is 8000 nucleotides only, base pair for DNA. And when it becomes RNA, obviously there is no pairing of bases, it is only single strand. So, 8000, 8 into 10 to the power 3, that is 8000 nucleotides. Out of that again mRNA, there will be lot of cutting and joining, and finally we get the desirable mRNA, that is only 2000 nucleotide. From that mRNA we will get our desirable protein. So, this is how the entire thing runs. Now, these are 10 hacks for more productivity. You can follow this. If you have any doubt, you can later on discuss also. Then coming to DNA sequence classes. What are these DNA sequence classes? They can be unique sequence DNA, that is they are not repetitive DNAs, they are very unique. And the second category is repetitive sequence DNA. So, as the name suggests, it will definitely be repeating itself. Then this repetitive and repetitive sequence DNA, they are again moderately repetitive or highly repetitive. More than half of this DNA sequences they are unique, that is single copy genes that code for proteins, fine. So, the first category is more than half, and repetitive sequence in human DNA at least 30% of the DNA sequence are repetitive. Now, this highly repetitive sequences, under this repeat sequences we have moderately repetitive and highly repetitive sequences. Now, coming to this highly repetitive sequences, in tandems of 5 to 500 base pair they will have repeats. Now, what is a repeat, what is a tandem? Repeat means the same sequence of the nucleotides will come again and again. So, such a repeat, if end to end joined, then it becomes a tandem. So, if you have observed, you have seen anywhere a double cycle, like two persons riding a single cycle, so that is called a tandem. So, like this tandem cycles we have tandem repeats. So, this tandem repeat will come again and again, you must understand. So, it is of 5 to 500 base pair repeats, and where they are found? Again in the dense region, that is the centromere and the telomeres. They are present in 1 to 10 million copies per haploid genome. Probably they play a structural role in DNA. That was about highly repetitive sequences. Now, moderately repetitive sequence, they are not clustered like we had seen in case of the highly repetitive ones, they are not clustered, they are interspersed in unique sequences, and less than 10 to the power 6 copies per haploid genome is present. Now, again there are two varieties, long interspersed nuclear elements and short interspersed nuclear elements, lines and signs. So, these are short notes for you, you will get questions on this. So, these repetitive sequences, they are moderately repeated and they have as per the interspersed areas, they are either long or short. So, and it is very interesting that they appear to be retroposons, that is they can jump from one site of the DNA to another site taking the help of an RNA intermediate. Okay. So, from one location to another location they can jump in the gene or the DNA, and they are known as retroposons. Okay. So, these lines, they are present in mammalian genome, they consist of around 20 to 50,000 copies of 6 to 7 kilobase pair lines. May I repeat? So, lines, they are of some 6 to 7 kilobase pair, that is 6 to 7,000 base pair, and there how many copies are there? 20 to 50,000 copies are there. these are the lines, the long interspersed nuclear elements, and signs are little smaller. They are 70 to 300 base pairs, and but they are more than 1 lakh copies per genome. They are more in number, but smaller in size. They are bigger in size and lesser in number, lines and signs. Now, this under this signs, there is a family of genes, that is ALU family, ALU. So, this they constitute almost 10% of human genome. Though it is a sign, it is almost 5 lakh copies per haploid genome is found, and it is highly conserved from species to species. Transposition is seen here, and that leads to a mutation which is seen in case of neurofibromatosis. So, transposition, I told you, jumping genes from one side of the DNA to another side with the help of RNA, that will lead to mutation, that will lead to neurofibromatosis. So, this is the case, this is a case of neurofibromatosis. In India also we see quite a lot of this disease. This is a genetic disease usually run in the family, and it is in very aggravating conditions, it is like this, but even people have only 4 or 5 nodes also in some places of their bodies, usually in the hands or in the back they will have this. So, sometimes in the entire face there may be lot of these neurofibromatic nodules, and it looks very bad also. So, this insertion of ALU family sign, if by transposition, that is by jumping, if it gets inserted into a gene, then the person lands in neurofibromatosis. Okay. So, in other certain diseases also they are involved, and particularly in transcription and splicing of RNA they are found in form of ALU B1 and B2. So, you may get a short note on SINE sign or the repeats, microsatellite repeats, that we will see now. As the name suggests, microsatellite, micro, small and distantly present, but they are also repeat sequences, smaller sequences, smaller patches of these repeats, they are found at different places. These repeat sequences that exist, that can exist both in grouped or dispersed manner. These most frequent ones are the dinucleotide repeats of AC. Now, this AC on one strand means what will be on the other strand? Obviously, it is complementary, means it will be TG. But apart from this AC short, I mean microsatellite repeat sequences, we may get CG, AT, CA, etc. So, maximum is AC, which is 50,000 to 1 lakh locations in the genome, and they are heritable traits. Since they are very much high in number and by dyes and all, it is easy to locate them by PCR. They are used for genetic linkage maps. Okay. So, these different diseases, they are used for studying the microsatellite polymorphism in different genetic diseases. Okay. Similarly, there might be trinucleotide sequences, and usually they are associated with genetic instability and hence lead to diseases. These trinucleotide sequences usually in the form of CGG, multiple repeats of this sequence will land in Fragile X syndrome, and Huntington's chorea, they have a repeat of trinucleotide repeat of CAG, myotonic dystrophy of CTG, spinobulbar muscular atrophy that of CAG. So, this is how trinucleotide sequences can also lead to some diseases. So, since I have mentioned here Fragile X syndrome, one of the diseases, they have this prominent long ears, a very long face. This is child's face, so the length is not seen, but adults, they will have a very long face, delayed development, mental retardation will be seen, delayed speech, large testes will be there, macroorchidism, that is, then hyperactivity will be seen, tactile defensiveness, gross motor delays, and autistic like behavior. They will be mildly autistic also. So, as per the expression of the disease, there may be some variation in the symptoms and signs. So, that was about the trinucleotide repeats. Now, there you may bump onto some other terms like pseudogenes. Now, these genes, they are processed by some means, but they will not encode for any functional proteins. So, usually the genes, they will be modified by any kind of crossover or recombination, whatever, but finally when they produce some protein, there may be abrupt introduction of some stop codon. So, functional protein cannot be synthesized. So, that is what is known as pseudogenes. Then as I told you about jumping genes, that was discovered by a lady scientist, Barbara McClintock, who gave the concept of jumping genes. So, this was a breakthrough and she also got Nobel Prize for that. So, this jumping genes are relevant, as we have just seen, in case of this neurofibromatosis and many other conditions also. Then there may be some other processing or changes in gene due to crossover, recombination, gene conversion, and they can change the genetic makeup, and if it continues generation after generation, they may have a role in evolution as well. So, these terms we must be aware of, and studying this phenomena, we have also found out how to manipulate them in the lab, so that we can get our desirable gene, desirable products, etc., and that has led to further research. There may be also sister chromatid exchange, and during just immediately post S phase of cell cycle, what happens? The DNA is replicated once and once only during the cell cycle. What are the different phases? G1, S, G2. In G1, it will be scanned for any anomaly in the DNA, it will be repaired, not able to repair, pushed to apoptosis. If it is rectified, it goes to the S phase. S means synthesis, there the DNA replication takes place, which we will take in our next class. And then this, when it is getting replicated, that means now 4 strands of DNA, they are almost very close, they have not separated altogether. Now, this is the time where there might be some crossover. Now, this sister chromatid exchange, usually if it is homologous, that is similar parts are being exchanged, then no change will be seen, but if it is heterologous, then it may lead to some kind of disease. Then another term, that is gene rearrangement. Now, this is especially seen in case of immunoglobulin. Immunoglobulin you have read recently, so it has 2 light chain, 2 heavy chain, then different portions you remember. So, this BL and CL genes, that is the variable region of light chain and the constant region of the light chain. They have their respective genes. Usually if you see the germline DNA, they will be far off, they are separated, but when they want to produce this IgG for the plasma cell DNA, they will come closer, but still they will be separated by an intron, and that is how it is by this gene rearrangement we can obtain our IgG. So, these are the few terms you must be very much well versed, because based on this subsequent classes will be taken. So, please go through the lecture again, and thanks for attending the class. Thank you.